Everybody. Welcome and coming on board and logging on and listening to what we do today. We've started off this back in 2019, the Australian Wildlife Week. I'm Steve Grabowski, the current president for the Australian Wildlife Society. So I've been on board now for 12 months, and it's a great honour to be able to assist with this meeting and run through it. There's a lot to cover. For your personal reasons, this video is being recorded. So if you're not comfortable with something, please raise your hand. We can mute it, but otherwise it's all going to be recorded. If anybody would like a copy of it later on, just let us know. We can pass it back to you. So again, welcome. Wednesday, the 2nd of October, 2024. This is the Australian Wildlife Week. We're celebrating Australian wildlife, which was established by the Australian Wildlife Society back in 2019. Australian Wildlife Week is to celebrate across Australia during the first week of October, every year, to encourage a positive relationship between humanity and nature. The Society aims to raise awareness of the wildlife conservation issues across Australia, inspire members and the community to develop a deeper understanding of these issues and implement wildlife conservation action to protect our native species, both flora and fauna. So, Wildlife of the Year. Before I move on to the format of today's webinar, I would like to highlight that the Wildlife of the Year is the greater gliders. And who watches the project on Channel 9? Raise your hand if you do. Anyway, how good was it? The project did a bit of a competition. They wanted to identify for 2024 what would be the marsupial of the year, and you wouldn't believe it, the greater glider won it. So the project on Channel 9 has picked the greater glider for 2024 as the marsupial of the year. So Google it, have a look. It's really good. We all thought maybe the koala would win it, but the koala came in second. So there you go. The powers that be that sit on this board have the insight to know and foreset who we should be picking for next year. So going through our gliders, our Guardians of the Guardian Gliders Parachute campaign, we contributed just over $10,000 to Wildlife Queensland for the new Greater Glider project. We encourage you to learn more about this amazing species, and more information is available on our website. It's going to get a great, there's going to be a lot of interest in the Greater Glider now, especially with the publication that's been done on Channel 9. Okay, format of the webinar. So today's webinar will showcase a breadth of wildlife research and conservation projects. I welcome Andrew Cox, CEO of the Invasive Species Council. He is our keynote guest today, and will be speaking first up. He will discuss the importance of action on invasive species to safeguard Australian wildlife. We will also be joined by 9 recipients of the Society University Research Grants and Scholarships for 2024. We will share the research on wildlife and suggest ways we can get involved with wildlife conservation. So a bit of housekeeping. Whilst we're talking, members are talking and playing videos, I ask that everybody keeps themselves on mute. A member of the society will introduce each other as we go through the format. If anybody stumbles, we're here to help you. Please submit any questions via the Q&A section. The keynote speaker will answer these at the conclusion of the talk. Megan will go through the questions and pick the ones out to read out. And once the questions time has approaches, Megan will organise that for us. The webinar will be a live stream on Facebook, and the webinar will also be recorded, as I've mentioned. So without further ado, I'll introduce our keynote speaker. I'm pleased to introduce our speaker, Andrew Cox, Chief Executive Officer of the Invasive Species Council, who will discuss the importance of action on invasive species to safeguard Australian wildlife. Thank you, Andrew. Over to you.
Thank you, Steve. Actually, I've got the wrong screen. Let me just share my screen. I want to start by welcoming everybody and wishing you all a happy Wildlife Week. What a great event. Thank you. I want to acknowledge I'm coming to you from Gundungurra and Direct Country in the glorious Blue Mountains, west of Sydney. I want to thank the Australian Wildlife Society for all of their fantastic work particularly the work they've been supporting the Invasive Species Council over a number of years. The Australian Wildlife Society were a critical supporter for our work on Kosciuszko to remove the large number of horses, feral horses in Kosciuszko National Park. So thank you to the Society, and also for supporting our work to remove feral deer from the Tasmanian World Heritage Area and the walls of Jerusalem. We're actually starting to see action on both of those campaigns now, and your support was really critical for us to get to that point. So thank you. But today I wanted to talk about invasive species. It is a critical issue if we want to safeguard Australia's wildlife. So let me just, I think, take you through some of the arguments and the evidence that's only just been recently compiled, really to demonstrate the scale of the impact they have on Australia's wildlife. So let me just move to this next slide. I guess many of you know, all of you know, the tragic story of many of the introductions to Australia. You know, many of them were deliberately introduced, like the rabbit introduced into Victoria for hunting, and the foxes and deer. Many species came just with all of our farmyard animals with the first fleet, pigs and cattle and horses, and many and then many were escaped from gardens. And prickly pear is a good example of where it was spread to all of Australia. And I guess this slide demonstrates the invasion process, that once something arrives, often there is little to stop it spreading. And so these become massive impacts. And we've got to, I guess, learn the lesson that once you introduce something, the consequences are broad and the impacts are high. And really the chance to stop it is before it ever arrives. So it's a big driver of loss in Australia. But let me explain a bit in terms of what it's causing, the damage it's causing to Australia. We've been doing some analysis about extinctions in Australia since colonisation. We know Australia's got a really bad track record on extinctions. It's actually quite hard to pin down exactly how many things are extinct at any one moment, because sometimes we might think an animal's extinct and then we find it decades later. And so therefore it's no longer extinct. And the ground parrot's a great example of that. I mean, there's good news there, but still we've got a tragic record of extinction. And particularly our mammals. You know, we all care here about our mammals. 43 mammals are now extinct. This is the worst extinction record for any country in the world. And the major driver for the mammal extinctions is invasive species. 39 of those 43 extinctions, the primary driver was invasive species, and mainly cats, number one, and number two, foxes. Cats are responsible for, at least solely responsible for just 25 of those 39 extinctions. So I guess the story here, I'll just take you through a few more stats, because this graph on the left shows total extinctions, but let's look at all the species that are extinct. There's, of all the species that are extinct, 81 species, 60 of those were primary drivers invasive species. That's 74%. For the mammals, I mentioned 39, that's 91% of extinctions caused by invasive species. For all vertebrate animals, these are the top 5 there. We have 54 species extinct. Again, 92% of those were caused primarily by invasive species. For the plant species, it's a different story, because for plants, the primary driver is mainly habitat loss and clearing. But 3 of those species that are extinct from the plants, just a quarter of them, are invasive species. And then in the invertebrate world, an important part of our animal kingdom, we had 10 species extinct. 3 of those were caused by invasive species. So the right-hand side shows that scale of the drivers. Sometimes you have multiple causes, but invasive species are by far the major cause of loss of our plants and of our animals and many plants. So that's what the latest science says. And this science is actually relatively new. If we pulled these figures together 20 years ago, we wouldn't have had such a clear picture of the threat of invasive species. And you might think this is an historical aberration for when a colonization first arrived. But if you plot these over time, the extinctions have been caused every decade. And the invasive species driver continues to be a significant contribution. So on average, we're losing about 5 species every decade. And of those 5, 3 are invasive species caused. And that trajectory doesn't look like letting up. The problem is going to continue if extinctions are the things we value. Obviously, there are other impacts as well, but extinctions as a simple record of our damage being caused, you can see the scale of the problem being caused by invasive species. Now, because I think it was sort of not well understood that it was a modern phenomena, many people thought it was an historical phenomena, extinction caused by invasive species. We commissioned a report by Tim Lowe and Carol Booth. There's Tim's a co-founder of our organization. Carol still works with us, but has been involved in our organization for 15 years. We looked at all the extinctions in the last 50, 60 years, since 1960. And you saw that graph, but based on some recent scientific papers, we've had 25 species become extinct since 1960. And of those, 17 were caused by invasive species. This is plants and animals. Now, of those 17, they're all animals. We've lost a few plants, not many, but you can see the scale of the impact even now. And also just to get you a bit of a handle on what extinction looks like in the modern world, these are the last 6 animal extinctions. Now, hopefully we all know these species, but most of you won't. I might start with the one on the right, the bottom right, the bramble K melames, a cute little mammal that we have lost from an island in the Torres Strait. It is our first climate change extinction. But the other 5 are all the losses were caused by invasive species. The top row, the Christmas Island pipistrelle, Christmas Island forest skink, and the blue-tailed skink, and this one here, the Lester's gecko, were all on Christmas Island. They are all now extinct, and probably because of the arrival of the wolf snake. It was a stowaway on ships. It arrived in the 1980s, and it slowly but surely wiped out these 4 species from Christmas Island. The white-chested white-eye is a bird only found in Norfolk Island, and it's extinct now, probably because of rodents and cats. So I guess my message is invasive species is a big driver, but they're not the only driver. We often have multiple impacts, threats, all impacting on often the same species. So they're overlapping threats, and we've got to make sure we're dealing with all of these big threats, whether it's climate change, the fire regimes, habitat loss, but invasive species. They all add up and work together to really force the decline of many of our species. And this is why we have over 2,000 species listed on our federal environmental threatened species list, because of these big drivers of loss. Now we might think, well, what's coming up ahead? We've done some work pulling together the papers of the imminent extinctions, not just threatened with extinction, but the extinction might occur within our lifetime. And you can see again, the major driver on the left is invasive species. I thought I might just highlight, because these comes from different scientific sources, within the next 20 years, a 50% chance that these species in the top are going to become extinct. Now, the big driver for the ones in red, these are the galaxias these are the small fish in New South Wales and Victoria, in streams, in coastal streams. The main driver of loss of those is trout predation. Trout has wiped out most of its these species in their catchment, and they're just hanging on in small sections of catchment, usually where the waterfall stops a trout going upstream, and the bit above the waterfall is where these galaxias are hanging on. So again, imminent extinctions. We've been trying to get action on the government. There was a bit of action after the bushfires because our fish are in trouble because of that. But still, there's barely any monitoring, let alone recovery efforts to protect these fish from trout. There's other things that are impacted by diseases like chytrid fungus, the frogs, orange-bellied parrot. I'm sure you know many of these species. They're on the brink. Now, further down, high risk within 10 years, these are the plants. We know habitat loss is a major problem there. But this bottom list are the species that are at risk from extinction, very likely to become extinct due to myrtle rust. This is a plant disease that arrived 14 years ago, and we think at least 16 species of the Myrtaceae family are going to become extinct in a very short period, just within once the adults die. We don't think there'll be any new trees. These plants will survive. It's a sad story. And what's driving all this? We're moving things around. We're breaking Australia's isolation by bringing species here which have never been in Australia, and the species in Australia have never evolved to adapt to. So it's trade, it's travel, it's all the importing of the goods, it's moving people around and the goods around, and either accidentally or deliberately bringing exotic plants and animals to, I guess, a naive population of animals and plants in Australia who are who lose out because they're not well adapted to those new species. I'll just speed up a bit because I guess this is a really difficult problem to solve, invasive species. And the simplest way to solve it in a sort of a methodical way, rather than just thinking, okay, we've got cane toads everywhere, let's just kill cane toads. We use this invasion curve to help us manage the problem. The invasion curve is a graph. This is assuming a species arrive. This axis along the bottom, the horizontal axis is time. So if a species arrived, like the rabbits, it slowly spreads to cover its full range of potential habitat. And then over time, as the area expands, the impact grows and the difficulty in dealing with it increases. And you get it to the point where once it's spread to a big enough area, it is impossible to eradicate. And then you've got to deal with biological controls. And that's where the feral cat and the fox and many widespread pests and weeds are in this area here. But the real opportunity is stopping them spreading or even eradicating them entirely. And you would have all heard about red fire ants. This is in the eradication phase. It's going to be costing billions of dollars to eradicate, but we still have a window of opportunity before it moves into this containment phase. So the earlier we eradicate, the easier it becomes. And by far the best approach is to stop it ever arriving in the first place. And I guess this is where we focus on biosecurity, protection at the border, stopping things arriving in the first place, having good surveillance preparedness, and rapid response plans. So that's the way our organisation has thought through about tackling this problem. And governments generally recognise this, but they're not very good at applying this. They're getting better. And our work is to try to make sure governments and everyone else thinks about this invasion curve in terms of responding to the threat. Our organisation was formed because of this book that was published around 1999, which was the wake-up call to conservationists who weren't working on this issue. Despite its scale, we're a standalone body whose sole purpose is to try to stop invasive species becoming to make it no longer the major driver of loss and degradation that it is. And that's largely through strong biosecurity. I'll just quickly I won't go into a lot of detail about how all the range of work we do, but we divide our work up into the 4 areas. The big system change to build up strong biosecurity within Australia so that we're actually stopping things arriving and we're dealing with the threats in a strategic way. Prevention and eradication are big benefits because that's the left-hand side of that invasion curve. And when you can controlling and containing things like horses and deer and cats, there's smart ways of doing it, and also focusing on protected areas. That's very important. And the other underpinning element we need is making sure we build social license, because often this work involves killing animals humanely. So we've got to make sure there's support for that. And that was one of the hardest things with feral horses. We've got to make sure we're involving the community and helping us mobilize support. We see a big potential for our Indigenous people to support this work because they manage so much land and they have a value system so aligned to what we're doing. That's how we work. And just an example of some of the wins we've had over the last little while, because we operate mainly doing advocacy, but also good analysis, we managed to secure from the incoming New South Wales government this range of measures simply because we put compelling arguments to the main political parties, and the Labor government agreed to this, and then now they're working through and implementing that. So we're really proud of some of the wins we've got. We're proud of the fact that we've got funding for fire ants, for example, for Southeast Queensland, which is going to impact all Australians. And so these are some examples of this, our recent successes. I'll finish up in what you can do, because everyone has a role to play. Obviously, we're trying to get the government to step up, particularly in that prevention space. But I think every one of you has a role to play. So we've got to think strategically about the invasion curve and using best practice tools. We've all got to be playing our part with looking out for things unusual. Or you would have known about the bird flu that's going to arrive in Australia shortly. If there's mass deaths or diseases, let's report that to the biosecurity authorities. Use iNaturalist to take a photo and send it into their national database. It's an amazing tool. And that if there's a biosecurity threat coming into that system, it automatically triggers a report. And also make sure you're not creating the problem yourself. Make sure you're not spreading weed seeds. Your vehicles are washed down after you go visiting anywhere, and you actually don't spread invasive species yourself. And of course, I'd encourage you to support our work, because we're donor funded and we've got a lot of work to do. So that's me. Thank you, Steve. And I don't know if you've got any time for questions.
Yeah, thank you, Andrew. And look, the Australian Wildlife Society, supports your projects, and we're strong advocacy for what you do and how you do it, and we totally agree. It's absolutely devastating with the invasive species coming out here and destroying what we have left. And instead of fire ants, we really need to get on top of that. But yeah, we do have one question, and I'm going to hand it over to our office manager, Megan, to coordinate that question and ask the question and take it from there. So thank you.
Okay. Feedback. Okay, let's try that again. We've got a little bit of feedback, but we'll persist. So we've got one question in the chat from Dr. Kumar saying, how can we control the extinction of species? And I'm thinking the extinction of native species from feral and invasive animals.
Yes, that's a great question, because unfortunately, many of these extinction trajectories were set in train when the invasive species arrived decades earlier, and the myrtle rust is a good example where we know these plants are going to become extinct in the wild, and there's very little we can do about it. And in that case, I think by making sure we've collected seed and maybe got some genetic material, so that if we can find ways of growing these species without the disease, we can still safeguard that or have some protected populations that are disease-free. So in many cases, it's going to be really difficult. But the best way, for a widespread pest like cats, we know that we've got to do cat control for feral cats. in the central desert in near Alice Springs, if we don't do cat control, we know that the desert rock rat would become extinct in the wild. So we've got to make sure we're supporting very targeted control efforts for the widespread pests. We need to be focused on biocontrol and technology to sort of come up with smart ways of controlling these widespread feral animals. And then we've got to stop the problem getting worse, and that's why that biosecurity is so important. So I guess you've got to be focusing on all parts of the invasion curve if we're going to start to lower that in extinction trajectory that's not looking very good.
Okay, thank you so much, Andrew. And you do have 2 more questions in the chat, but because we are at 10:56, if you wouldn't mind just answering them in the chat, if that's okay.
I'm happy to do that, Megan. Thanks very much. And I might
Great.
pop on about 10 minutes later too. Sorry,
Thank you so much. Okay, so I'll now ask Patrick to introduce our next speaker.
Come on, give me the clue. Am I back on?
You are, yep, you're introducing our next speaker.
Okay, thank you. It's my pleasure. My name is, I have to let Suzanne in. She locked herself out. It's my pleasure to introduce the next speaker, George Lester from the University of Melbourne. He's going to speak to us about one of the current problems in relation to climate change and the effect on stainless bees. So it's my great pleasure now. And he's the winner of our revered past Vice President, Dr. Clive Williams, the award, and George is the winner of our 2014 award, and we're looking forward now to hearing George tell us all about it. Thank you, George. Over to you.
Thanks very much for the introduction. So yeah, here's me in the research team, and like you said, I'm from the University of Melbourne out of the Mayfield lab, and I'm studying native bees. But before I begin, I'd like to acknowledge the lands of where I'm talking from, the lands of the Wurundjeri people. And in the theme of Wildlife Week, the translation of Wurundjeri actually translates to both the manigum tree and the witchetty grub. And the Wurundjeri people are known as the witchetty grub people. So that's just a fun fact about that. And I'd also like to acknowledge the lands of where my research take place, the lands of the Yolngu First Nation. And I can draw parallels between both of these First Nation groups because they both use bioindicators to signify the calendar seasons. Like in Melbourne, we see that we're in the season of Porneet, which is signified by the flowering of murnongs and dianellas. And similar in the season of Willamuru in Arnhem Land is signified by the finishing of the flowering of goodacres or stringy barks, as well as the opening of seed pods of the Willamurus. And so here's my research in a nutshell. Firstly, the Yolngu First Nation have been noticing recently some concerning declines in the honey harvest of native stingless bees. So they've asked us to go up to northeast Arnhem Land and look at native bees, and how we've been doing that is analysing the pollen on each of the native bees to see what flowers that they're visiting. And then we're also looking at the flowering times of these key feed plants, which have also been reported to be shifting the flowering phenology of the flowering plants in the area. So that's the summary, and here's where the research is taking place, right up in the top of northeast Arnhem Land in remote Northern Territory in the Eucalyptus tetradonta stringy bark woodlands. And this is a really remnant, remote part of the world. And to put into context just how remote it is, the area of northeast Arnhem Land is about the equivalent of the country of Belgium. And prior to our research taking place, there was less than 50 native bee or any bee records in the whole region. So there's really no baseline data whatsoever to go off. And so what we've been doing up there is we've been doing lots of plant surveys and looking at the different flowering plants in the regions. And we've been sequencing the DNA of these plant species to make these unique barcodes, which are like sort of indicators of each of the different species, like barcodes are. And so this comes in handy then when we collect the bees and are looking at native bee pollen diets. We can sort of scan the DNA of the pollen that the bees are carrying, and that'll give us an indication of the different flowering plants that the native bees are collecting pollen from. And we're building these networks which connect all of the native bee species in the area that we've collected, and see what different flowering plant species that the bees are relying on and using as feed plants and their key resources, and sort of identify the ones which in particular are really, really depended on by the native bees in the area. And this comes in handy because then we can look at the different important key resources to these native bees, and we can sort of assess whether the flowering times of the flowering plants of the key resources is something that's impacting the native bees. And we've identified a few trends, like the top left corner is the stringy bark or the Gadeka, and this seems to be flowering later and later in the season as the years go on, whereas there's other species which seem to be flowering earlier, and you can understand how these all being out of whack can be having impacts on native bees in the area. And what we're doing with this information is we're relaying this back to our research partners with the Yolngu First Nation, and this can help to inform their management strategies in the future, as well as engaging in a longer term monitoring project so that we can get a better idea of what's going on with the honey declines in the stingless bees through all of this research. And yeah, it's been a really awesome opportunity to work with the Yolngu people on this, and I'm really grateful. And yeah, I'd like to also thank the Wildlife Society for the funds. So thank you.
Thanks, George, for that. That's great. Hence, this is why you received an award from us. You're doing exceptional work, exceptional job, and we look forward to continuing seeing you thrive. Do we have any questions? We have questions for George? We've got. No, we're going straight through. Okay, I've been told we're going straight through. So, unfortunately, our next director, Robin Chisholm, is unable to make it here today. So I have the.
I was going to ask the question, Mr. Chairman. I'm going to ask the question. In relation to the flowers that you were monitoring appear to be shrubs rather than trees. There's a open forest woodland up that that comes into season. Yeah, the range of native trees up there that flower prolifically when they come into season. But the flowers you were monitoring appear to be grass cover, lower shrubland, yeah, which is subject to fire regime from time to time, which complicates it.
Yeah, absolutely. And we're following the methods of previous research that's been done on DNA metabarcoding of flowering plants. So we're trying to get more of a replicate study, and also the things with like the remoteness and the inaccessibility of the area that hinders what sort of equipment and things that you can take into the area, and the processes like that. That's one thing we've definitely would like in the future, is to look at the eucalypts of the area as well and do some canopy surveys as well.
It's a difficult area to get to. I spent 3 months in Kakadu many years ago on birds.
Sorry, we're just over time. Can we refer some questions to the chat, and we'll just keep moving on? Sorry about that.
Okay. So our next guest speaker is Natasha Bartels. She's from the University of Technology, Sydney, UTS scholar. We're going to talk about coral, the conversations we need to have. Can coral communicate with each other via chemosignaling under stress? So over to you, Natasha.
Hi, everyone. I will share my screen. Okay, has that pulled up presenter view or is that just the full slide for you guys?
Perfect.
Perfect, thank you. So my name is Natasha Bartels. I'm a final year PhD student in the Future Reefs research team as part of the climate change cluster at the University of Technology, Sydney. Before I begin, I'd like to thank the Australian Wildlife Society for providing the funding for this project. It's one that I'm really excited about and what I'll be talking about today. So I'll be discussing coral conversations, investigating how corals communicate via chemosignaling. Before I get stuck in, I'd like to acknowledge the traditional custodians of the land on which I live and work, the Gadigal people of the Eora Nation. I pay my respects to elders past, present and emerging, and I acknowledge that these lands always were and always will be Aboriginal land. So you've probably, well, I imagine everyone's probably seen on the news this year, a little bit earlier in the year, we had another mass bleaching event on the Great Barrier Reef. So it turned it from this beautiful, vibrant, colourful seascape to something a little bit more like this in very large areas of the reef. So we have bleaching and dead corals. It's a bit of a barren wasteland, not a lot can survive there. As many coral species are only found on the Great Barrier Reef, as it continues to face mass bleaching events, we're increasingly running the risk of losing entire species, whole areas of reef, and the inhabitants of those areas. So as you can probably imagine, it's something us coral scientists are talking about quite a lot. But something I didn't anticipate before I started this project is that corals might actually be talking about it themselves as well. So the idea of corals communicating with each other is something researchers have actually been discussing for a very long time, but just haven't been able to pin down, especially in relation to coral spawning, which is the remarkable event during which corals release sperm and eggs into the water. It's the only opportunity for sexual reproduction for most species, and it only happens on a single night each year. We've long known that there's several environmental factors that influence the timing of spawning, but earlier in my PhD, I did actually find that corals also rely on chemical communication to synchronize their spawning down to a window of time of as little as 10 minutes. So they're basically sending out these chemical signals to each other, saying, hey, it's time to release your sperm and eggs. Let's get fertilizing and make some coral babies. And so that got me thinking that can't be the only time corals are communicating with each other via chemicals. So I'm now looking at if corals communicate with each other under stress. So during marine heat waves, the water often gets warmer at the surface first, and then we start to see a depth-dependent gradient of bleaching. So like you can see, it's a great example in the photo, the coral at the top is the most bleached, and the one down the bottom is the least bleached. And so what I want to know is if maybe that coral at the bottom has survived just a little bit better only because the water hasn't been quite as warm as at the top. That's definitely a factor. But is it possible that it's also receiving warning signals from that coral up the top? And so it's able to ready itself before the heat wave even reaches that deeper depth. And then on the flip side, could the chemicals released by the coral at the top actually cause harm to their deeper neighbors and compound that stress? So we can answer these questions by using an experimental system I've developed with the funding provided by the Australian Wildlife Society. And to give a very, very brief overview, this system basically allows me to capture the chemicals released by a coral under high temperatures, take those chemicals and then dose them to a second coral that's being held at just ambient or normal growth temperatures. Then after a few weeks, I'll take that second coral and subject it to heat stress and see if it fares better or worse for having received those chemical warnings from that first coral. And apart from the idea of corals chatting with each other being really cool, both outcomes have some pretty considerable implications for management strategies on our reefs. So if one coral or a stressed coral negatively affects its neighbours, coral gardeners who grow corals on nursery frames for restoration purposes might want to consider increasing the spacing between corals on nursery frames. But on the flip side, if corals do kind of warn each other of impending stress and those corals deeper down perform better, conservation efforts might then pivot to manipulating those chemical cues before predicted heat waves even happen as a way to get corals to better ready themselves as temperatures increase. Positive or negative, understanding the impact and reach of chemosignaling between corals could be really important to include when modelling bleaching and recovery, as those models are what we use to show us where we need to focus our efforts. So it's pretty timely research considering the mounting threats our reefs continue to face, and it could provide a really promising new avenue to explore for safeguarding our reefs. I kind of like to think of it, this method is Google Translate for corals. So corals are speaking in chemicals, and with this method, we now have a way to understand them. I know I've gone a little bit over time, so I imagine there's not going to be time for questions, but if you'd like to reach out online, I am on Twitter. I would love to hear from you and keep chatting, and thank you for listening.
Thank you, Natasha. Does anyone sent through a question? Okay, so, yeah, you're right, Natasha, no questions. But please, if you do have any questions later after the meeting, send them through. We'll get them out. So now it's my turn. I'm delighted to introduce our next speaker, Rose Lowndes from Western Sydney University, who will speak about bogong moths. Over to you, Rose Lowndes.
Great, thank you for the introduction. Can I check that everybody can see me, hear me, see my screen? Great, that's all good. Hi, I'm Rosie. I'm a PhD candidate at Western Sydney University. I'm coming to you today from Darug country, and I'm studying the joy that is bogong moths. So this is a bogong moth. They're quite small little fellows. They're only 2.5 centimeters, about the tip of your thumb, and fairly ordinary in appearance. But these moths perform one of the most extraordinary migrations every single year. They're born across breeding sites in southern Queensland, inland New South Wales, Victoria, and South Australia. And every year they migrate towards the Australian Alps, where they are dormant over the hot summer months before flying back to their breeding sites. For some of these moths, this is a journey of over 1,000 kilometers, and they do it all within one generation. Bogong moths are extremely important within their mountain summer habitats. They're crucial to the nutrient cycles within the mountains. They provide around 7 tons of nitrogen and 1 ton of phosphorus every single year. They're also a really important source of protein for lots of animals in the Alps, including the critically endangered mountain pygmy possum and also the southern bent-winged bat. They are also culturally important to many First Nations Australians. Unfortunately, in the last few years, people haven't been seeing so many bogong moths. In 2017 to 2019, it's thought that only 5% of the expected 5 billion bogong moths arrived in their mountain sites. This may be due to lots of factors. There was a drought, which is thought to have caused really inhospitable conditions in their breeding sites, but also factors like agricultural intensification and climate change as a whole have probably affected these incredible insects. A really important thing to consider when trying to plan effective conservation measures for bogong moths is their migratory routes. We know where they are during the summer while they're in the mountains, but a lot of the rest of the year, we're not quite sure where they are. So plotting out their migratory flyways, the paths that they take to and from the mountains, is a really crucial thing to research so that we can conserve them in the best way possible. And that's what I'll be studying for my PhD. Thanks to the funds from the Australian Wildlife Society, I was able to do my first fieldwork for my PhD. I actually only came back yesterday. as we're thinking of several different ways to map out their migratory routes. One of those is looking at the genetics of the bogong moths and also looking at stable isotope analyses. So the nutrients that bogong moths take in as larvae, there are specific isotopes that change throughout the Australian landscape, and they remain present in bogong moth wings as adults. If we find a bogong moth that's performed its migration already and then analyze the isotopes within its wings, we are able to discover its natal origin, where it was born. And using that information, we can track where they have flown from and try and find out the main flyways that different groups of moths take. During this field work, we were collecting plant samples for calibrating the stable isotope analysis so that we can make it as specific to bogong moths as possible. We also were trying to trap bogong moths so that we could have a look at their genetics. We looked everywhere really from Sydney until Dalby in South Queensland, and unfortunately we weren't able to find any bogong moths. A lot of the farmers we talked to up in South Queensland talked about how dry it's been. They hadn't had the spring rains yet while we were up there, and it's thought that that might have really affected their time of emergence and even the survival of bogong moth larvae up there. It's so crucial for us to know where bogong moths are at different types of the year, and that's why we're really promoting people use the app iNaturalist to record any bogong moth sightings that they've seen. People can also use the moth tracker by Zoos Victoria, who are really promoting trying to see where bogong moths are at different times. Thank you very much. If anyone wants to ask any questions, feel free to in the chat. And thank you for listening about bogong moths.
Thank you, Rosie. Great speech. Once again, it was good to learn and how amazing a little moth like that can be so important to the system. Brian Scarsbrick is unfortunately not with us at the moment, so I'll introduce the next speaker. So I'm delighted to introduce our next speaker, Matthew Rose from the University of Tasmania, who will speak about the collapsed habitats that threaten Australian marine wildlife. I'm very interested in this one. Matthew, over to you.
Cheers, Steve. Thanks for that. So my name's Matt. I'm based down at the University of Tasmania. I'm going to give a pretty rapid fire overview of my research relating to Australia's two great reefs that are under threat. So Australia is home to the Great Barrier Reef, stretching about 2,300 kilometres. But we also have the Great Southern Reef, which stretches approximately 8,000 kilometres, wrapping around the south coast of Australia from Kalbarri all up to central New South Wales. And these are home to really diverse communities that I'm sure we all love, and hence why we're here today sharing our knowledge. But they are under threat from a number of stresses, largely anthropogenic. And so we need to understand what dynamics are threatening their persistence and what dynamics can promote their persistence. So that's where all of my research interests have kind of led, is to understanding what determines an ecosystem state and then how do we manage from there. Oh, my animation's not working. Apologies.
Still with us there, Matt?
Yes, still with you. Apologies for that. I was my animation's not working for me. My PowerPoint isn't showing on the screen.
You might have to use your facial expressions.
About that. We'll try it in. Can you still see my screen?
Yeah, it's back up now.
Okay, cool. Thanks for that. It's not working on the actual slideshow, so I'll just do it from here. So my research is largely interested in understanding reef functions. So reef functioning is the movement of energy and matter within an ecosystem. So we get energy and matter moving into a ecosystem through sunlight. Habitat forms such as corals and kelps assimilate energy into matter, providing food and shelter for a range of species that exist there. These species then have a feedback effect on the habitat itself through their own feeding processes. And so there's really complex dynamics evolving between habitat and the species that inhabit them. And my research is really interested in understanding the movement of energy between these within these links. So kelp removal is one ecosystem function that I'm interested in. This is the process of herbivores feeding on kelp and removing it from the system. And we really, in functional ecology, we really want to understand how many kilograms of mass are being removed per meter squared per hour to determine the variable contributions of kelp removal by different species, understanding each individual species impact, and then obviously understanding the combined impact, which is important towards dictating the ecosystem state. Now, this is particularly important in Australia with these two great reefs under threat. So there's intense warming occurring quite rapidly right across the East Australian coast. In Southeast Australia, indicated by the red arrows on this plot on the left, on the figure on the left, there's actually a hot spot of ocean warming, which is accelerating to up to 4 times faster than other regions in the world. And with all of this warming, we're having species redistributions occurring. Some of them could potentially be benign. Some of them are incredibly problematic. One of the classic examples is the long-spine sea urchin extending its range to Tasmania and the negative impacts that that has had. So on the right here, we can see that with these anthropogenic stresses of warming, we're seeing shifts. We're seeing habitat states shifting from high habitat to these low collapse states dominated by turfs. These are comparatively less complex, offering less niche availability for various species, and then obviously has implications for what communities could exist there. And so once these shifts has occurred, the species within the system play a really important role in determining the recovery trajectory. And that's why we need to be identifying the rates with which species are feeding and removing and shifting material around within the system to determine how they're influencing those recovery trajectories. And that's where my research comes in. So right across Australia, our team has been applying a bunch of camera methods, survey methods, and assay methods. So putting out portions of algae and portions of squid, and then seeing how much is consumed after 24 hours. And then from camera work, counting bite rates of various species. And we're doing this right across a gradient of depth, temperature, habitat state, so we can understand how the dynamics of species feeding influences that habitat state. And this work has fortunately been supported by the AAWS, so thank you so much.
Good morning, folks. My name is John, and I am a director of the Society. I am delighted this morning to introduce our next speaker, Luke Florence from La Trobe University, who will speak to us about mycorrhizal diversity in Australian temperate forest. Thank you, Luke. Take it on from here.
Cool. Thanks for the introduction. So today I'm going to discuss the temperate forest ecoregions of Australia. I'm going to also discuss the concept of nitrogen deposition, a little bit on the ecological history and function of mycorrhizal associations, and I'll give you a really brief overview of my project. So the temperate forest ecoregions of Australia are home to outstanding examples of global biodiversity and evolutionary history. They encompass the Gondwanan rainforests of Australia, which represent the largest patch of subtropical rainforest on Earth, with the highest diversity of ancient Gondwanan plant lineages. They're also home to the tallest flowering species on Earth, the mountain ash, However, despite their ecological significance, these forests face climate change challenges, including drought-induced tree mortality, and furthermore, situated in the areas of highest population density in Australia, these forests are also threatened by nitrogen deposition. So what is atmospheric nitrogen deposition? It's a process whereby environments such as our forests are indirectly fertilized by human activities such as the burning of fossil fuels and industrial agriculture. Research in the Northern Hemisphere has shown that nitrogen deposition can alter the composition of forestry species, and that these changes are often mediated by the mycorrhizal partners of the trees below ground. And furthermore, drought can exacerbate these nitrogen effects. However, these Northern Hemisphere paradigms are likely not to be reproducible in Australia, where we have a unique environment and biodiversity. So what are mycorrhizal associations, and why should we care about nitrogen deposition effects? So when I go through my weekend walk through the mountain ash forests in the Yarra Ranges, the tree roots of these mountain ash below the ground. They're colonised by mycorrhizal fungi. These fungi form complex below-ground networks that absorb nutrients from the soil as well as water to form an association that's not only essential for the health of over 95% of all tree species, but it's also essential for life on land. So there's two main types of mycorrhizal associations. One type is so ancient that it predates trees, and it played a critical role in the terrestrialization of plants some 450 million years ago. This mycorrhizal type is so persistent that it's endured the 5 big mass extinction events. So I'll introduce you to arbuscular mycorrhizal fungi. This ancient lineage is particularly important for the mineral uptake of nitrogen for trees. So mineral nitrogen is the type of nitrogen that we fertilize the environment with through atmospheric nitrogen deposition. Because of their affinity to mineral nitrogen, arbuscular mycorrhizal associations, they tend to gain a competitive edge under high nitrogen deposition scenarios. Unlike arbuscular mycorrhizal fungi, ectomycorrhizal fungi are only about 160 million years old. And while still impressive, they've only endured a single mass extinction event. These fungi share an ancestral history with decomposing fungi, and therefore some of these groups have retained the capacity to access organic nutrient, which is otherwise unavailable to trees and arbuscular mycorrhizal fungi. So nitrogen deposition can alter the mycorrhizal dependency of these ectomycorrhizal trees to render them not so necessary for nitrogen uptake. Many of our Australian trees, such as eucalypts, they form this type of association. And this association is particularly important for drought tolerance. And so, therefore, as nitrogen can weaken these relationships, that could ripple on to also make these ectomycorrhizal trees more susceptible to drought. So, so my research aims to advance our understanding of mycorrhizal fungal diversity in Australia through a collaboration with the Australian Microbiome Initiative. I've sampled and attained 140 temperate forest soils where I'm doing metabarcode analysis, much like George was doing with his bees. So as we speak, these soils are being sequenced thanks to the funds of the Australian Wildlife Society. And with these data, I aim to map patterns of below ground diversity, evaluate the impact of soil nitrogen and aridity on this diversity, so they're my nitrogen deposition and drought proxies, and also to estimate the relative vulnerability of mycorrhizal fungi to nitrogen deposition and drought proxies. And I'm really excited to contrast what we find here to these paradigms that have been developed in the Northern Hemisphere. I'd just like to finish by acknowledging the Wurundjeri people of the Kulin Nation on whose land I work and meet with you today, and also to the funding bodies and collaborators who made this research possible. Thank you.
Thank you, Luke. Very impressive. I appreciate your time. So our next guest speaker is Charlotte, I'll say this right, Rigo Lott from the University of Newcastle. who will speak about understanding koala population dynamics to inform conservation management initiatives. So over to you, Charlotte. Did I say your surname right?
Yes, it's Rigolot. Yeah, that's right.
Beautiful. You got the floor.
Okay, so I'll try to share my screen. Can you see my screen in full screen?
No, we can't, unfortunately.
Oh, you can't. Okay. Should be right now.
Yes, it's working.
Okay, great. So yeah, thank you for the introduction. So hi everyone, I'm Charlotte. Before we start, I'd like to start by acknowledging that I'm presenting today and working on the traditional lands of the Pambalang clan of the Awabakal Nation. And I'm conducting my field work on Warimi country. I wish to pay my respect to elders past, present, and emerging, and to any First Nation people present here today. So my PhD project focused on understanding koala population dynamics to inform conservation management interventions. So to begin, let's discuss the broader context. So as you all know, and as we heard before, biodiversity is declining globally, putting wild population at risk of extinction, especially in Australia, where mammals are heavily affected. The koala is a highly specialized arboreal species and is declining due to habitat loss, disease, domestic dog attacks, and climate change-related bushfires. So in 2022, the status of the species changed, and the koala is now endangered in Queensland, Australian Capital Territories, and New South Wales. So while several conservation actions have been proposed, their impacts on populations are not well understood. So the study of these actions and long-term monitoring of wild populations, both before and after implementing conservation actions, are essentials. So in New South Wales, to efficiently use the funding toward koala conservation, we need to obtain information on wild populations. Well, this is where this project comes in. My PhD aims to study several koala populations across different habitats in New South Wales national parks to analyze their risk of extinction and the major threats they face before implementing any management action. So by studying these populations for several years, before and after the implementation of management actions, we aim to compare the positive or negative impact of different management methods on the population movements, the structure, and also the risk of extinction. So to obtain these data, we are employing several methods. We use drone thermal imagery at night to locate koalas in their habitat and to have a good understanding of their abundance and distribution. So for these surveys, we are working in several national parks on the north mid-coast of New South Wales. Thanks to this technology, we were recently able to catch and collar 20 koalas in 2 national parks. And we also equipped them with Bluetooth ear tags, which are basically more of a backup in case of any koala is dropping. In addition, we deployed camera traps in these national parks to monitor predator presence and activity. So using these data, I am building models to assess the risk of extinction of these populations in function of different scenarios and by integrating different management actions. So these models will incorporate population dynamics, carrying capacity, diseases, genetics, or vegetation type layers, for example. Another key focus of this PhD is to analyze the movements, behavior, and interactions of these koalas to understand the current status of the population. So we use GPS tracking data to examine home ranges and habitat use, as well as to evaluate population structure and individual connections. So we are also investigating potential prey-predator interactions and dynamics, as for example, we have currently no evidence of dingoes' predation on koalas in these national parks. We want to have a look at how prey and predators species use and share their habitat. So as mentioned earlier, we fitted 20 koalas with collars across 2 national parks on the mid-north coast in July. So far, we have collected 2 months, a bit more than 2 months of data, and we are just having a first exploratory look at the data. So after a full year, we will examine movement data such as differences in travel distances, based, for example, on sex and vegetation, along with other covariates. So currently we are examining the movement patterns of individual koalas as shown on these maps. Given that it is now breeding season, koalas are starting to move quite a lot. So we need to wait a few more months before having a look at home ranges. We also plan to collar more koalas. We also plan to collar more koalas in this population. And I just want to add that during the first 2 months of our study, we regularly tracked and cited these koalas, and none has showed any sign of discomfort or issue related to their collars. So to conclude, we hope that this project will bring comprehensive and extensive data on New South Wales koala population dynamics and behavior prior and after conservation management actions, and as well as insight into the benefit of such actions. I would like to express my gratitude to the Australian Wildlife Society for their generous scholarship. Thank you very much for supporting this project.
Thank you, Charlotte. Very good presentation. So I'm delighted to introduce our next speaker, who just so happens is celebrating a birthday today. Jasmine Towell, from the University of Adelaide. We'll speak about climate change, impacts of marine invertebrates, and over to you, and a happy birthday to you from all of us.
Thank you, Steve. Just share my screen. Can everybody see that?
Yes.
Cool. Awesome. So my name is Jasmine. Thank you, Steve, for the introduction. I'm a PhD student at the University of Adelaide. Just a quick acknowledgement of country. I'd like to begin by acknowledging the traditional custodians of the land on which we gather today, the Kaurna people, and pay my respects to their elders past, present, and emerging. So temperate intertidal ecosystems in Australia have a unique biodiversity, hosting more endemic mollusk species than anywhere globally. Intertidal marine invertebrates, like the ones on the screen here, play crucial roles in coastal defense and provide food and habitat for a range of species. However, their distribution and habitat availability is at risk due to climate change. Unprecedented rates of environmental change, such as rising ocean temperatures, fluctuations in salinity, and wave exposure, lead to uncertainty of species survival under such threats. The intertidal zone is where the ocean meets the land between low and high tides. It has 3 different zones, as demonstrated here, the low, mid, and high zone, depending on the tide. Intertidal zones host a unique trophic system and species interactions which drive biodiversity. Intertidal species are perfect candidates to study climate change impacts, as they already experience a wide range of stresses and conditions in their day-to-day life. They have to deal with exposure, tidal fluctuations, and are exposed to different extremes of the elements constantly. This has led to them developing key adaptations to cope with living in their extreme environment, which can be studied to understand how species are responding to increasing climate change. So this is just sort of an overall figure of my PhD work. To mitigate climate change threats, we require knowledge on how species are responding and will respond to climate change. This project aims to identify phylogeographic barriers and adaptive traits of intertidal marine invertebrates in temperate Australia. Using this, we will explore how climate change-induced environmental changes are affecting biodiversity and predict future range shifts of species. So just to clarify a bit of the jargon there, phylogeography allows us to see how populations have responded to past environmental change, which we can then compare to current studies to understand how populations are responding to contemporary climate change. And the morphologies, so it's a bit of an example down here, of species control different functions that affect their fitness and whether they adapt to their surrounding environment. For example, a limpet could have increased foot size to account for increasing wave exposure so that they can better stick to rocks. Australian Wildlife Society funding has allowed me to examine the following research questions. The first has past, i.e. Pleistocene climate change affected gene flow between marine invertebrate populations across temperate southern Australia, and are there current barriers to gene flow? And secondly, are there morphological size and shape differences in shape within species across existing environmental gradients that may allow us to predict how species will respond to future change? Starting in mid-October this year, I will be conducting fieldwork across temperate Australia to sample genetics and morphology of 6 invertebrate species. These are on the screen here. So we've got the waratah anemone, the tall-ribbed limpet, rough-beaked mussel, purple four-plated barnacle, little blue periwinkle, and the black-fingered crab. These species were all chosen as they have similar life history characteristics, such as long larval dispersal on a mostly sessile adult stage. So the genetic and morphology data will be analysed with the environmental data to correlate which environmental variables may have driven past population division and current morphological trait variation. So mentioned earlier, the environmental data is wave exposure, sea surface temperature, and salinity. So a bit of an example again is the foot size of the limpet. So hypothetically, under low wave exposure, limpets may have a smaller foot size as they don't have to hold on to their living surface as strong. And under increasing wave exposure, as you see here, due to climate change, their foot size may grow as it tries to adapt to increasing wave exposure. So many of the sites are remote and have not been studied, including sites across the Great Australian Bight here and the Coorong. This project will utilise a multidisciplinary approach using genetics, morphology, and ecological methods to develop innovative and robust strategies to combat worsening climate change. This project and its expected outcomes of climate change mitigation would have not been available without funding from the Australian Wildlife Society. So thank you very much. Thank you for having me to speak today, and thank you everyone for attending as well.
Thank you, Jasmine. And yeah, your talk is very intriguing about the foot sizes there and responding to climate change. So it's my pleasure to introduce the next speaker, and that's Alexandra from the University of the Sunshine Coast, speaking about male tiger sharks. Take it away.
Hello. How is everyone's good? Let me just share my screen. Share. Okay, awesome. So yes, hello, my name's Alexandra. I'm with the University of the Sunshine Coast, and I'm working on tiger sharks based along the east coast of Australia. This is a bit of an overview of my entire PhD, and so I have a bit of a wide focus when it comes to tiger sharks. I'm looking at their overall movement over here on the far left, using a variety of tracking tags. I'm also looking at behaviors using some video camera tags. I'm looking at diet, doing stomach dissections, and then I'm also doing diet work as well with eDNA using swabs. The focus of what I'm doing right now does evolve around my satellite tags, and so that's what I'm going to be speaking to for the next few minutes. So you can see here I have 2 different tag types. We have the splash tag, which will get placed on the dorsal fin of the tiger sharks that I catch. And then I have my pop-up satellite tag, which actually goes here along the side of the dorsal. And they each capture a variety of information that produces maps such as below, essentially looking at how they move throughout the water column, where they're moving, time of day that they're moving. We collect environmental factors with this as well, such as water temperature. And from that, we're able to get a really good idea of what these sharks are doing in the water, where they're going, and hopefully we can figure out why they're actually moving to those different areas. So, like I said, it's the east coast of Australia, which is just up here. So I work all the way up from northern Queensland down into southern New South Wales. And yeah, so when it comes to the tiger sharks, I'm not just going out and fishing for any tiger shark. I am focusing on mature males. Why males? Well, it's because from the data that we do have for tiger sharks along the East Coast, almost all of the paper publications that we have don't include mature males in their analyses. They might have captured some and tagged a few, but they've tagged too little to actually include them in any of the analyses and any kind of conclusions that they make about movement of tiger sharks. So my focus is to directly kind of go to different areas along the coast where we have an idea that the mature males like to hang out, so we can actually tag them and get data from them. A lot of different species have sex-biased movement patterns that has an effect on biology and ecology for both the species and the surrounding area. So being able to really round out the information we currently have on adult females and juveniles of both sexes will be adding the mature males into that. So that's why my focus is the mature males. What I will be doing, thanks to AWS, is going out on boats using both smart drum lines in association with the New South Wales DPI, as well as rod-based fishing to fish for tiger sharks, get them alongside the boat, tag them with both of these tags, and then send them off on their way. And then when we do get the information, it looks something like this. So on the left here, you can see these are the pop-up satellite tag tracks for 6 different sharks that I've been able to get tagged so far. And as you can see, there's a wide variety, but compared to the females and juveniles over here, we can already start to see some small differences in between movement patterns. So hopefully, after this next fieldwork season, with a couple more tags put out, we'll be able to make some really good comparisons between the sexes and come up with any maybe sex-biased movement, biological hotspots, etc., based on the data that we're collecting. So that's my project. I'm really excited to do some more field work in a couple of weeks with AWS's support. Yeah, that's my project. Thank you.
Over to you, Patrick.
What was that?
Alex, it's great work. I really take note that Hayden likes to go exploring.
Yes.
Vanuatu's nicer waters over there.
Yes. No, they like to go really, really far. It's quite phenomenal.
Yeah. But it is interesting how they do congregate. They don't go too far. They go far, but they seem to hang around one side of the coastline.
Yeah, and what's really interesting is we have a lot of work in terms of Australia on the west coast, and tiger sharks along that coastline of Australia. And so it'll be really fun once we get all the data collected to compare the two coasts and see if we have differences in just our area of Australia.
Thanks very much. I don't know what's happened to Patrick, who was introduced next speaker, so I'll do it for him. So I'm delighted to introduce Rebecca from Edith Cowan University. She'll be speaking about bedagongs. So Rebecca, are you with us? Yes?
Yeah.
Over to you.
Cool. Is everyone able to see my screen?
Yep, that's it. You've got the floor.
Cool. So hi everyone, I'm Rebecca. I'm a PhD candidate at Edith Cowan University, and I'll be chatting about a study I'm undertaking to look at resource use and availability for 2 threatened bettong species that are planned for reintroduction to Darkatuk Island. But first, I'd like to acknowledge the traditional owners of the lands in which I'm presenting on, the Whadjuk Noongar people, as well as the traditional owners of the following areas where my sample collections were undertaken. So broadly, my PhD is looking to improve the translocation success of Mycophagus or fungus-eating mammals. Mycophagous mammals play an important role in ecosystems by serving as fungal dispersers when they and bioturbators, as well as when they dig for underground fungi. But unfortunately, many of these mycophagous mammals are threatened in Australia. So translocations are often used as a conservation tool, but they come with many challenges, including but not limited to selecting the right habitat with suitable resources. So this particular study forms 2 of 5 experimental chapters in my PhD and focuses on the reintroduction of booties and woylies onto Dirkartok Island in the Sharp Bay World Heritage Area of Western Australia. So booties and woylies are Mycophagous bettong species listed respectively as being vulnerable and endangered by the EPBC Act. A little bit of context about Dirkartok Island is that it was historically managed as a pastoral lease grazed by sheep and goats, and feral cats were also established on the island. So the combined effects of this led to the loss of 10 native mammals from the island, including booties and woylies. Fortunately, today the island is designated as a national park, and initial threats to these mammals have been eradicated. So the Return to 1616 project was established to translocate 13 species, including booties and woylies, back onto the island with the goal of species translocation, species conservation and ecological restoration. Despite having initial threats addressed, the island's landscape has been altered. So how would we know if fungal resources required by the bettong still remain? So in this study, I plan to use environmental DNA metabarcoding techniques on scat and soil samples to firstly gain a better understanding of the diet of booties and woylies, and I'll do so by identifying the fungi being consumed and the fungi available to these bettongs in their existing populations. So using environmental DNA techniques again, I plan to undertake a survey of the fungi available on the island to determine if there are suitable resources for bettongs there. And lastly, I'd like to determine if the 2 species are likely to compete in a scenario of limited resources on the island. My sample collections have successfully been undertaken in autumn and spring this year over 6 sites. This was made possible by Department of Biodiversity Conservation and Attractions, as well as Australian Wildlife Conservancy, that provided in-kind support for my fieldwork and made sure that I had all my samples I needed. Woylie scats are collected from Mount Gibson Sanctuary, Dryandra Woodlands, New Haven Sanctuary, and booty scats are collected from Foray Island, Madawa Karara National Park, and New Haven Sanctuary during trapping programs. And soil samples were collected from an existing booty and woylie population each, as well as on Dirkartok Island. So the next steps for me will be to undertake lab work, and this will use the funding awarded by Australian Wildlife Society. Very briefly, that includes DNA extractions, PCRs, library preps, and sequencing. That'll be followed by bioinformatics and stats that involves demultiplexing sequences, processing them into units that can be identified by reference to fungal sequence libraries, and then stats will help me to identify species richness, diversity, and overlaps in fungi between booties and woylies, as well as between locations. So at the end of this project, I expect to have a better understanding of booty and woylie diets, including their ability to adapt across multiple environments. I also expect to have an understanding of fungal assemblage on Dirkartok Island and its capacity to host the bettongs. And ultimately, I hope that the knowledge gained from the study will be able to guide the reintroductions of booties and woylies into the island, as well as inform future conservation decisions for the species.
Thank you, Rebecca.
Happy to take any questions in the chat.
Yeah, we'll take questions after. That's very informative, and we hope we get that research completed. We've just a little bit over time, so we are right to our last speaker, and I'm sure Natalie Garcia from Murdoch University, she will discuss with us today whether fragmentation alters predators' diets. So Natalie, your last speaker, you've got the floor. You can finish it off with a bang.
Cool, thank you very much. Can you see my screen?
Yes, we can.
Cool, beautiful. Yeah, hi. So I'm Natalie Grassi. My PhD is with Murdoch University, Bush Heritage Australia, and the Department of Biodiversity Conservation Attractions. And one of my chapters of my PhD is looking at whether fragmentation alters predator diets. So just first off, acknowledging I'm presenting to you all from Whadjuk Noongar country, but my fieldwork and all my samples have been collected from Coorang Noongar country, about 6 hours out of Perth. So as we've kind of had the introduction to today, feral cats and red foxes are obviously really significant issues in Australia for our wildlife. They've contributed to over 20 extinction events, and it's unfortunately very likely that other species are going to follow suit soon. We know so far that both of these species predate roughly 400 vertebrates each, and a lot of what we do know about that comes from diet analyses. It's as glamorous as it sounds. Basically, you dissect either a scat or a stomach to figure out what they're eating. It is a little bit gross, but it is also really, really important for elucidating trophic interactions, identifying the species which are most at risk, and helping to prioritize conservation actions. Unfortunately, feral predators are not the only threat we have facing our Australian wildlife. As I'm sure we all know, habitat loss and fragmentation is another really, really big issue. So fragmentation specifically is the process in which a large continuous landscape gets broken apart into small isolated patches. During that process, you experience habitat loss, where natural structures like trees or shrubs that native animals would have once used for escape or avoidance are removed. Unfortunately, these barren landscapes are a preferred hunting ground for cats and foxes. To combat this, there's obviously been extensive revegetation, but that takes a lot of time to develop into something that's more complex and actually offers those escape or avoidance structures. So this brings me to my study site. So I'm working in the Fitzsterling landscape, which is pretty much everything between the Stirling Range and Fitzgerald River National Park. Confusingly, within that, you have the Fitzsterling macro corridor. So that's that little strip shown in orange. Then you've got the coastal Palenut corridor just below that. So that's in green. And that's a really nice, healthy, intact landscape. Unfortunately, the Fitzsterling is not quite so pristine. So over the recent decades, it has faced a 90% loss of its original vegetation, mostly for agriculture. It's then had a lot of revegetation, which, while I can't overstate the importance of that, unfortunately, it's resulted in this mosaic of remnant habitat, farmland, and re-veg, which creates a lot of ecotones, which are the barriers between two habitat types and another preferred hunting ground for cats and foxes. So these sorts of landscapes are really vulnerable to predations, and we need to be able to focus our efforts here, which is why I've been looking at fox and cat stomachs. So far I've cut up 67 cat stomachs and 76 fox stomachs. My cats were all leg hold trapped by Bush Heritage and DBCA, whereas my foxes were shot by some volunteers. What we found so far is there's really not too much diet overlap between the predators. So cats generally prefer to eat native mammals, birds, and invertebrates, whereas foxes were preferring invertebrates, introduced mammals, and our native mammals. When you kind of get a bit of a closer look into these, the predators or the prey items that were most heavily associated with cats were generally smaller size, so native and introduced rodents, rabbits, honey possums, pygmy possums, and birds, whereas foxes were generally eating more invertebrates, vegetation, sheep, and kangaroos. So important to note here is obviously the size difference between prey items. Cats are not really known to scavenge, they'll only hunt live, and they generally prefer to eat small but more frequent meals, and so a lot of these animals were caught live. In contrast, foxes are known scavengers, and a lot of these prey items were actually eaten as carrion that was predated in farmlands. So based on this, we've kind of found that in this landscape specifically, cats are actually the bigger threat to native wildlife and should probably be the focus of more of our conservation and management outcomes. Of course, what we want to know as well is how fragmentation is altering diet. So the way we want to do this is to see how diet varies with a percentage of remnant and revegetation cover around the point of sample collection. And then the other thing we want to do is compare how my diet analysis results compare to those from the Fitzgerald River National Park, which were analysed by my supervisor, Sarah Comer. So the funds which have been generously provided by AWS have been used so far for fieldwork. Unfortunately, it's a very long way from Perth to the Fitzsterling. It's about a 1,000 kilometre round trip. And as glamorous as cutting up stomachs is, it does take a lot of sample jars and a lot of ethanol to store things. So that's where the remaining money is gone. Massive thank you, of course, to the Australian Wildlife Society and to all of my volunteers who have been able to make sample collection possible. We obviously don't really have time for questions today, but please feel free to ask in the chat or email me if you need to. Thank you.
Thank you, Natalie. Yep, and it was a good finish for the end of the season, end of the term. So I'd like to thank everybody. Thank you again for attending the Australian Wildlife Week webinar. I'd like to make a special thank you to all our speakers today. Wonderful presentations. Apologies for the time that we have a full agenda. Maybe next year we might extend it for an extra half an hour. Special thank you to our keynote speaker, Andrew Cox. His valuable time is precious and we appreciate him coming on board. We hope the webinar has inspired you to explore and develop a deeper understanding of one or more of the wildlife conservation matters discussed here today and take action to preserve our wildlife and nature ecosystems. If you'd like to find out more about today's projects, how to support us and to help support these and other important conservation efforts, or have any other further questions, the links to the webinar and occupying information will be made available on our website, which is www.aws.org.au. I'll be there soon, so please drop us an email at info@aws.org.au if you've got any other specific questions. Finally, if you are not already a member of the Society, we would like to encourage you to become a member for as little as $55 for a year. Your support is valuable to us, to our survival as well, and with greater numbers, we're then able to then support you guys in your projects so that you guys can develop better strategies to implement laws, I suppose, or changes to the systems and way things get done so that we can make a big impact and continue to preserve our natural fauna and fauna. So thanks, guys. Appreciate it. And that wraps it up. Bye.